Hi, everyone. This is episode 732 of People Behind the Science. I'm Dr. Marie McNeely, and today I am excited to be joined by our guest, Dr. Ralph Dewey. This episode is made possible with support from our sponsor, New England Biolabs. Listeners, are you curious how molecular biology is moving forward around the world? Hear from the scientists who are driving discovery and expanding applications of existing technologies on the New England Biolabs podcast, Lessons from Lab and Life. Host Lydia Morrison interviews revered scientists working in genetics, synthetic biology, diagnostics, molecular cloning, and other fields. Get ready to explore the cutting edge of science and learn what these remarkable scientists have to offer. Don't miss out on the opportunity to hear exclusive stories about the world of laboratory research. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And today, our guest, Ralph, will be talking more about his own life and research, listeners. So get ready to meet another one of our fantastic people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Ralph Dewey. So, Ralph, welcome to our show today. How are you?
I'm doing very well. Thank you.
Well, Ralph, we are so excited to have you with us today, and we're looking forward to learning more about you and your work. But first, let me start by telling our listeners a little bit more about you and your background. So, listeners, Ralph is the Philip Morris Professor of Crop and Soil Sciences and Adjunct Professor of Plant Biology at North Carolina State University. He was awarded his bachelor's degree in biology from Utah State University, followed by his master's and PhD in crop science from North Carolina State University. Afterwards, Ralph received an NSF postdoctoral research fellowship in plant biology to conduct postdoctoral research at the Waxman Institute at Rutgers University. He joined the faculty at NCSU in 1991, and he has been issued 34 U.S. patents for his discoveries in plant biotechnology, with several more pending. And Ralph was awarded NCSU's Philip Morris Endowed Professorship in 2009 for his research on harm reduction in tobacco. So, Ralph, we're excited to get to know you as a scientist, of course, in this conversation today, but also as a person. So, can you start by telling us what you like to do when you're not doing science?
My favorite thing I like to do outside of work is that whenever my wife and I have a weekend that's free is to enjoy it at a beach condo that we have. It's about a 3-hour drive from where we live. Also, my wife and I have always enjoyed college sports, particularly football and basketball. And now as our son and daughter both attended different universities as undergraduates and now as postgraduates, as my daughter's currently in medical school and my son in law school, the number of schools that we find ourselves rooting for just keeps growing. Mm-hmm. Right now, we're keeping up with teams from 5 different universities.
Oh, wow.
And finally, about 4 years ago, I began writing a novel, and I found that this to be a rather enjoyable pastime.
Fantastic. Can you give us any teasers about the novel, or is it secret for now?
Well, I can give a brief overview. In its broadest sense, it's about science, religion, and nuclear holocaust. So the original concept started years ago when, for reasons I can't even recall, I asked myself the question, if the cryptic events recorded in the book of Revelation really were a description of some type of apocalyptic end of the world, what would that look like in today's world of science and technology? So the two main characters are a cyber technology genius who has come to believe that he has been ordained of God to protect those going to follow him from the calamities that are about to befall the earth, and a university professor who has become unwittingly aligned with this so-called prophet. Central to the story is a catastrophic nuclear disaster that destroys a major portion of the U.S., rendering it uninhabitable because of the high levels of radioactive contamination left behind. Now, the only survivors of the disaster are pockets of these presumed cult communities who were able to survive because of the research conducted by the professor and his team. So the professor and ultimately the whole world is confronted with having to decide whether this prophet character is legit, or alternatively, was he the one responsible for causing the disaster? So that's kind of it in a nutshell.
I am intrigued, Ralph. You'll have to let us know when the book is out.
I will.
It sounds like you've got a lot of different passions outside of science. You mentioned, of course, going to the beach, rooting for all these college sports teams, and this writing passion as well. But I know you're doing some really phenomenal science in the lab. So, Ralph, how do you describe your science to someone who's not in your particular field or maybe not in science at all?
Well, in simple terms, I would say that I use the tools of molecular biology to identify and characterize genes of agronomic importance in crop species, and when possible, alter those genes in a manner that will add value to the crop above and beyond that which can be accomplished using traditional breeding approaches.
Very interesting. And I know much of your work, Ralph, has focused on tobacco. So can you talk a little bit more about the importance of doing this research on tobacco?
Yes, I'd be glad to. I think that our projects in tobacco, in fact, are among the most interesting and worthwhile that we've conducted. First of all, to put things in proper context, everyone's aware of the hazards of smoking, and now it's still the leading cause of preventable death in the U.S. And thankfully, there's been enormous progress in smoking reduction, and the number of people who smoke has been on a steady decline, but it's still a major problem. Now, in a perfect world, the problems related to smoking could be solved if everyone just quit, but that's not the reality we live in. So despite all of the restrictions that have been placed on smoking, the constant educational messaging, and even financial disincentives imposed by the increased taxes on the product, there are still a lot of people out there who either willfully choose to smoke or have found themselves in a position where they can't seem to quit. So therefore, it's important where possible to find ways that one can decrease the hazards of smoking, even if incrementally. So say, for example, you came up with a way to make smoking less hazardous by something as little as 1% to 2%. Considering the fact that in the U.S. alone, there are still over 400,000 smoking-related deaths each year, that 1% to 2% reduction in harm could translate into helping thousands of people. So as a molecular biologist, I work with the genetics of the plant. And if you ask yourself the question of what can be done to reduce the harm caused by tobacco products at the level of altering plant genetics, the answer, quite honestly, is not a lot. That's because the vast majority of toxicants found in cigarette smoke are the products of combustion. That is, if you take any plant, not just tobacco, dry it down, burn it, breathe it in, you'll be exposing yourself to the majority of compounds that are causing cancer and other illnesses in smokers. There are, however, a small number of carcinogenic compounds that are only found in tobacco because these are ones derived from the alkaloids that are unique to the tobacco plant. Alkaloids such as nicotine and its closely related derivative nornicotine. These alkaloid derivatives are carcinogenic compounds that are not products of combustion and are unique to tobacco. Now, the most important of these compounds are called tobacco-specific nitrosamines, or TSNAs. And of these TSNAs, the worst ones are ones referred to as NNN and NNK. Numerous studies in animal systems have shown that NNN is highly associated with cancers of the esophagus. NNK is one of the main carcinogens associated with lung cancer. These are compounds we actually can do something about at the level of genetics. And over the years, my tobacco breeding colleague Ramsey Lewis and I have come up with several genetic strategies that have been shown to be effective in greatly lowering the levels of these carcinogens in tobacco products, both in cigarettes and in smokeless products like chewing tobacco. So another major goal of our tobacco research has been to use both molecular, genetic, and traditional breeding tools to develop tobacco plants that produce negligible amounts of nicotine. The motivation for doing this stems from numerous studies and clinical trials that have been supported by NIH and the FDA that have shown that for many smokers, transitioning for a period of time to cigarettes whose nicotine levels are too low to sustain an addiction response can actually help wean them off of their habit and quit. So ultra-low nicotine cigarettes can thus serve as a smoking cessation tool. In fact, in December of 2021, the FDA granted a company that I've been working with called 22nd Century Group the authorization to market very low nicotine cigarettes as modified risk products that can help you smoke less. In fact, the words helps you smoke less is in bold letters on each pack. So the tobaccos we've developed and are continuing to develop are being done to support this effort.
I think this is really interesting research, Ralph, and I think so relevant today when we're starting to think more and more about all the health risks of these products that we're taking into our body. So I think it's really exciting to have this research going on that has the potential to have a really big impact for a lot of people, and that can be inherently motivating and inspiring. But do you have a quote or a saying or something that really keeps you motivated when times are tough in science?
Yeah, I like a fairly well-known quote found in the Robert Louis Stevenson essay, El Dorado. I believe this quote is most effective when you include a couple of lines prior to it, so that's what I'll do here. And it goes like this. O toiling hands of mortals, O unwearied feet, traveling ye know not whither. Little do ye know your own blessedness, for to travel hopefully is better than to arrive. So I like this quote because conducting research is in fact a journey of hope. Now, the type of research projects conducted in my lab, once a new project's initiated, it takes a minimum of a year and sometimes 3 or 4 before we know whether it'll be successful. And many times it isn't. In fact, I can give you more examples over my career of projects that failed to meet expectations than those that did. But in the end, none of them can be called total failures because with each project, win or lose, you learn something that makes you a better scientist. So to me, what makes this career the most satisfying is being continually engaged in a journey of hope. Research is an exciting journey. I think sometimes we get so focused on the end result that we don't fully appreciate the privilege of just being able to participate in the journey itself. That's why I like that quote.
Absolutely. And I think a lot of scientists out there can relate to those lines of toiling hands and weary feet and just those long hours in the lab, putting in your all to get to this point years later, like you said, in some cases where the project comes to a complete point. So I think that's phenomenal and a good way to keep yourself motivated, kind of thinking about this big picture, the hope that you're working for. And I know it can be helpful to have these motivational and inspirational figures as a scientist as well. These people that you look up to who have perhaps made it to where you'd like to be, or these people who inspired you to pursue the path to begin with. So when you think about your own career, Ralph, are there examples of mentors or role models or just these historical figures that you've really looked up to?
The first name that instantly comes to mind is that of my PhD advisor. I was very fortunate as a graduate student to end up working in the lab of Sam Leffings was his name. He was one of the pioneers in the field of plant molecular biology, a member of the National Academy of Sciences, and he laid much of the original groundwork in the area of plant mitochondrial genes and genomes. He was not only super intelligent, but also rather large in stature. He had this reputation of showing little patience for fools. So many graduate students in the department found him to be quite intimidating, myself included when I first joined his lab. Once he concluded, though, that I was halfway competent, I really couldn't have asked for a better mentor. It was during my time as a PhD student that I really gained my passion for molecular biology, and a huge part of that was a result of my interactions with my mentor. He was working in the Leffings lab when I first experienced this excitement of science being an adventure, this exploration into the unknown. And his office was connected to the lab, and many times I'd be working my lab bench and I'd hear his bellowing voice shout out, "Dewey, get in here." He'd call me in just because he'd either discovered something very interesting in a research paper he was reading, or he'd learned something relevant to our project from a colleague over the phone, or simply because he wanted an update on the latest experiment I was working on. He was also incredibly supportive of promoting my development as a scientist. One year, for example, he was invited to give one of the keynote talks at a plant molecular biology themed Gordon conference, but instead he gave a slot to me, a graduate student. I was only one of 2 graduate students who spoke at that meeting. So to me, there's no doubt that Sam Leffings shaped my development as a scientist more than anyone else.
Oh, I think that's amazing. And I think having those mentors can really transform your career. And I'd love to rewind a little bit. You mentioned you had this phenomenal experience, of course, as a PhD student, but what got you to that point? Ralph, what really got you interested in science to begin with and started you on this path?
So I came from a very academic-oriented family. My father and 3 of my uncles were all PhD-level scientists. 2 of my uncles were professors at Utah State University, where I did my undergrad. In fact, there was one semester when I was there that I was taking classes from both of my uncles.
Oh, wow.
One taught plant breeding and the other statistics. My third uncle was a marine biology professor at UC Davis. But the biggest influence by far was my father, who worked for the USDA. He was a research geneticist who studied rangeland grasses. Growing up, I always thought that his job was so interesting, especially during the times he would go on these plant collecting expeditions in far-flung regions of the earth. Like he spent most of the summer in Iran in the 70s before the revolution that happened there. He also went to Russia and China and Inner Mongolia, and he would go to the very remote regions of these countries to collect grass species that didn't exist in the U.S. and bring them back to be included in plant collections that the USDA was maintaining. So even though there were no expectations that me or my siblings go into science, but just being raised in this type of environment, when I entered Utah State as an undergraduate, I did, knowing from the very beginning that I wanted to go on and get a PhD and become a researcher, though I initially didn't know what type of research I wanted to do. That is until I took a course in molecular biology. Now, that was the most interesting course I had ever taken up to that point in my life. and it made me want to pursue a PhD using the techniques of molecular biology to study plants. From there, I went to NC State for my graduate studies. As I just mentioned, worked on projects involving identification, characterization of genes located on the mitochondrial genome of maize, with a special emphasis on identifying a gene responsible for a trait called cytoplasmic male sterility, or CMS, which is a trait of great importance to the hybrid seed production industry. After receiving my PhD, I did a postdoc at the Waxman Institute at Rutgers University in New Jersey. And in addition to gaining more training and experience professionally, it was at Rutgers where I met my wife, Pat, who was a graduate student in the same lab. So from Rutgers, I went back to NC State when what I considered to be the perfect job opened up in the crop science department. My future wife was a PhD student at Rutgers, but when we decided to get married and I got the job at NC State, she left Rutgers with a master's degree, transferred to NC State, and finished her PhD there. So that's the story of how I ended up to where I am today.
I think that's wonderful. And what was it like for you then, going back to NC State and now being on the faculty side of the equation?
It was great. Like I say, the circumstances were right. I love this area of the country in North Carolina. And I was already familiar with a lot of the people at NC State. So NC State, there are some institutions that won't hire their own, so to speak. I am fortunate that they didn't have that attitude in my department. So I was happy to have the opportunity to actually come back and work as a faculty member in the same place where I got my PhD.
Very cool. I think that can be helpful because you sort of already have some of these connections and you already know a lot of how the school works and operates. And I think that's a big learning curve for people who are coming into a new institution to start their own lab.
Yes, that is a definite advantage.
Well, we touched on your research earlier in our conversation. I'd love to circle back and dig into some of the details here, Ralph. So is there a particular project that you are working on at the moment that you are just the most excited about and want to tell me and our listeners about in more detail?
Right now, I'm very excited about the newest project in my lab involving mitochondrial genome editing. Like we already touched on briefly, say the mitochondria has its own genome that encodes a small number of genes that are distinct from the main genome of the nucleus. One of the unique characteristics of mitochondrial genes is that they are not inherited in the same manner as nuclear genes. In the great majority of plant and animal species, the mitochondria are passed strictly through the maternal parent. Take us, for example, half the DNA that's found in our nuclei came from our fathers, and the other half came from our mothers. The genes in our mitochondria, however, came 100% from our mothers. Most plant species are the same way. The difference in inheritance is important in agriculture because of the way hybrid seeds are produced commercially. Now, for listeners who may not be familiar with this area, one of the most time-tested and proven ways to maximize yield in most crop species is to take two very different inbred lines and cross them. The offspring of this cross will grow more vigorously and be higher yielding than either of the parental lines. This phenomenon is called hybrid vigor, and currently over half of all major crops cultivated worldwide are hybrids. To produce hybrids on a large scale, however, can be tricky because in addition to the two parental lines that were picked being able to cross with each other, they can also self-pollinate, giving you the same inferior parent all over again. So this is where the trait cytoplasmic male sterility, or CMS, comes into play that I mentioned before. Around 100 years ago, researchers observed naturally occurring mutations in plants such as maize that prevented them from shedding pollen, and these mutations were only passed along maternally. Later on, researchers like my PhD advisor went on to prove that these CMS genes were located in the mitochondrial genome, thus explaining their maternal inheritance. Now, without getting too deep into the weeds of how hybrid seed production systems works, suffice it to say that these CMS genes solved the problem of how one can produce large amounts of hybrid seed. So even today, these CMS mutations serve as the cornerstone for producing hybrid seed in many crops, such as rice, sorghum, sunflower, sugar beets. The problem is that naturally occurring CMS systems either don't exist or don't work very well in many crop species. So what we did in my lab very recently was to show that we could create new CMS traits by taking an essential gene from the mitochondria, converting it to a nuclear gene that expresses in all parts of the plant except those responsible for pollen development. Then we used genome editing tools to clip out and eliminate the original gene from the mitochondria. This resulted in a CMS phenotype where the plants could not produce pollen. And most importantly, because the mutation we created resides in the mitochondria, it's inherited maternally and thus can be useful in producing hybrid seeds. Also, during the course of our experiments, we fortuitously discovered a new way to produce a seedless fruit phenotype, much like you see with seedless watermelons and grapes, where only small, soft seed remnant is left behind. Now, we did all these experiments using tobacco as the model system because of how easy it can be genetically manipulated. But in theory, there's no reason to think that we couldn't successfully use the same strategy in other species to produce CMS systems for hybrid seed production for crops where the trait would be beneficial, or seedless fruits in cases, again, where that would be the end goal.
Absolutely. And Ralph, I think if it hasn't sunk in for listeners, the implications of this work are huge. The fact that the system that you've developed could be applied to a lot of different plants of agricultural significance is tremendous. So to give our listeners a sense of the scale, the scope, or the potential impact, could you comment on that?
Well, yes, we are very encouraged by our results and the potential implications, but our enthusiasm does have to be tempered by the fact that we are still at a very early stage of the research. And there is this big gap between observing promising results in a model plant species grown in a greenhouse and going all the way to showing efficacy in a relevant crop species in the field. So for the seedless fruit applications, we have recently introduced our system into tomato, which is a species closely related to tobacco. So we didn't even have to change any of our constructs. Now, right now, we have several very small tomato plants growing in a growth chamber in the basement of my building, and we're excited to see what will happen when they start to set fruit. Even tomato, however, would still represent a proof of concept step, as there already exist ways to make tomato plants that produce seedless fruit. If our tomato results show that our technology can, in fact, work in a true fruit species, then we'll try moving on to a fruit where customers would like to have a seedless option, but currently don't. One example would be blackberries. Many consumers, myself included, don't like eating blackberries because of those hard, gritty seeds that are annoying to chew and get stuck in your teeth. There would be a lot of interest in a seedless blackberry. Another fruit important to North Carolina is muscadine grapes. Muscadines are currently very important to the wine industry in the state, but one of the main reasons they haven't caught on so much as a table grape is because of their large seeds that have to be spit out. So for the CMS hybrid seed application of our technology, once we've made a couple of more tweaks to the system in tobacco, we'd like to test it out in rice, which is a species that can be easily transformed and where hybrid seed is important to the industry. So there's still a lot of work to do, and I'm sure there's going to be challenges to overcome before we can declare success. But for now, I'd say that we're proceeding cautiously optimistic and looking forward to taking it through to the next steps.
Well, Ralph, this is a really exciting project. And listeners, if you love hearing about cool science, there is another podcast you may be interested in. New England Biolab's podcast, Lessons from Lab and Life, is your source for staying informed about the most up-to-date molecular biology breakthroughs. Host Lydia Morrison interviews prominent scientists and delves into their research to uncover how their findings affect society in meaningful ways. Tune in to learn from the experts and get a glimpse of the cutting-edge advancements being made by scientists around the globe. Don't miss out on this opportunity to gain new knowledge and stay informed on the latest discoveries. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And Ralph, of course, you hinted that there have been challenges for you. And I know it's not always smooth sailing in science. And we love talking about these challenges because I think people don't always get to hear about them. They just see this finished product. So, Ralph, do you have an example of something that you really struggled with, or maybe a major failure that you had that you could tell us more about and talk about how you worked through it?
The most notable struggle I've had in my career dates back to my postdoc years. Now, I just come off an amazingly successful PhD experience, better than I could have ever dreamed of, having resulted in first author publications in Cell, Science, and PNAS, and among others. By the time I'd then moved to New Jersey to do my postdoc, again at this Waxman Institute in Rutgers, in the lab of Dr. Dan Klessig. Now, this research in Dan's lab focused on characterizing the molecular mechanisms by which plants use to protect themselves against pathogen attack. And that was what I went there to work on in order to broaden my experience and skill set. So I had no intention of doing anything related to the mitochondria when I began my postdoc. But after I got there and saw the resources and the technical expertise that was in Dan's lab, as well as that of the neighboring lab, I thought that all of the resources were there that could enable one to overcome one of the biggest obstacles in mitochondrial research, which was the inability to transform the mitochondrial genome. That is, to incorporate an expressive foreign gene within the mitochondrial genome as opposed to the nuclear genome. Now, such an innovation would have represented a huge technological breakthrough. So I pitched the idea to Dan, my postdoc advisor, and he let me work on the mitochondrial genome transformation project. Now, I will always be appreciative of Dan for giving me the freedom to do this, especially given that it was unrelated to any of his research projects, though I'm sure that there were many times later he regretted this. Anyway, I spent 2 years hammering away at this problem, going back and forth between experiments where I'd get these encouraging glimpses that our constructs were genuinely expressing within the mitochondria versus experiments where the results were completely negative. In the end, nothing proved to be repeatable, and Dan and I mutually came to the conclusion that we just needed to cut our losses and move on to something else. So there I was, 2 years devoted to a project and nothing tangible to show for it, as we had nothing that was publishable. Fortunately for me, soon afterwards, the job opportunity at NC State came up, and I was able to take advantage of that and move on before my career went totally down in flames. Say, being a postdoc, this isn't a period of time where you can go all that long without having, say, publications to show for it. But the whole irony of the situation is that I left Rutgers being a far better scientist than when I arrived. And yet on paper, there was really nothing to document this. And even now, I can still console myself somewhat by the fact that to this date, there still have been no reports of anyone being successful in transforming a plant mitochondrial genome. I don't think they've been successful in doing this in animal systems either.
Oh, very interesting. So was there ever a moment in that 2-year dry spell where you thought about giving up or just sort of changing fields or leaving science altogether?
No, that thought never crossed my mind. I always had the attitude that things will work out, even if it means moving on to different projects, that ultimately things will work out.
Gotcha. Well, thank you so much for talking us through this tough time. It sounds like making that difficult decision to walk away from the project, even after you had invested 2 years, was a smart choice. I think sometimes you do have to just call it and move on. And I think that way you can go on to these successes in science and you can celebrate them. And I think it's so important to take a moment to celebrate all of the wins in science, whether it's a big win like getting a new job there at NCSU, or whether it's a small victory, just a little thing that finally works in the lab. So do you have a favorite success story, Ralph, that you can tell us about?
Yes, I do. And I think the success story that I would like to share is the story behind the one experiment that more than any other of the course of my faculty career has had the biggest impact in shaping and defining my program. In fact, we're going to kind of pick up my story where I just left, going from being a postdoc at Rutgers then to a beginning faculty member at NC State. As I mentioned before, I really do have a dream job here at NC State because of the freedom that has allowed me in pursuing projects that I find are interesting and worthwhile. But this didn't really necessarily start out perfectly. Again, one of the reasons I say my job was unique was the crop science department is what you'd refer to as, they say, a commodity-based department where faculty members are typically hired to work on a particular commodity, whether it be corn or wheat or peanuts, for example. And my position was different when they created this. Again, back in the early 90s, they wanted to bring in someone who had expertise in these new molecular biology techniques, not to necessarily bring their own project, but to work with other faculty members, particularly plant breeders, where these kind of techniques could help advance their programs. And so it gave me a lot of freedom to work with different projects on different plant species because I wasn't tied down to any particular commodity. Now, again, while this sounds great, at this point, it didn't necessarily start out that smoothly because one negative aspect about stepping into a position where they didn't want the person to necessarily bring along an existing project is it can be a little difficult to convince funding agencies to give you money when you have no previous history in that particular area. So the first couple of years, all I had to work with was a couple of small startup grants, and my team consisted only of me, a technician, and one graduate student. So most of my original research associations were with a soybean breeder and a couple of soybean physiologists who were working on improving the quality and abundance of the oil within the seed. So for those who may not be aware, soybean oil is by far the major vegetable oil produced in the U.S. And in addition to food applications, it's an important feedstock for a number of industrial products such as plastics, resins, and the production of biodiesel. But there's a lot of room for improvement, however, in the oil profiles that currently exists. And altering the genetics of the plant is an effective means for changing and improving the quality of the oil for a given application. So back in the early 90s, when I started my faculty job, there was very little knowledge about the genes responsible for oil production, not just in soybeans, but in any plant species. The first step was to simply identify the genes encoding key steps of the pathway. At that time, we didn't have all the genomics resources available today, so identifying these genes was far from trivial. So working with the soybean physiologist and the breeder, I'd tried numerous approaches to identify some of these genes, but over the course of a 2.5-year period, I was unsuccessful. But now from the literature, I was aware that researchers, and actually this was a group at Duke University, had identified one of these pivotal genes in yeast. And I thought that a good approach would be to try identify the equivalent soybean gene by complementing the yeast mutant that this lab had created. What made this experiment particularly tricky, however, was that the yeast mutant displayed no growth phenotype. So the only way one could determine whether they had successfully complemented the mutation was by using this labor-intensive, technically difficult procedure called colony autoradiography. Now, another problem I faced was that a critical component of this experiment was a radioisotope that was very expensive. It cost around $4,000, which was even more money back in the early 90s. And I simply didn't have the money for this. So I went to my physiologist colleague, hat in hand, and asked him whether he'd be able to buy this for me. Graciously, he said yes, and I am forever grateful for him for doing so. So my lab started these experiments, which involved transforming the mutant yeast strain with a soybean gene library we had made and plating out the yeast on dozens of large petri dishes, each containing thousands of yeast colonies, then transferring these colonies to filter paper, incubating them with this radio-labeled isotope, which the experiments, if successful, would involve incorporating the isotope into the yeast membrane, where after we dried it down, put X-ray film on, then we could visualize it. Now, we could screen around 250,000 colonies in each round of experiments, but after 3 rounds, we'd still found nothing. And I only had enough of this expensive isotope left for 1 more round of screening. And at that point, I'd actually made the decision that if this final screening attempt failed, that I would need to give up and move on to a different area of research because we were just getting nowhere. But then it happened is on that final experiment, one lone dot showed up on one of the many X-ray films, and it turned out to be the gene we were looking for. Now, that discovery led to my first USDA grant, which then led to an NSF grant, and so forth and so on. So it's really no exaggeration to say that every notable success that my research program has had can ultimately be traced back to the fact that that one experiment ended up working. If it hadn't, I can't imagine what would have happened to my research program, or if I would have even been able to get tenure. So over my entire career as a faculty member at NC State, that one successful experiment early in my career was clearly the most impactful.
Oh, wow. Did you realize at the time when you were kind of running that last batch that everything hinged on that? Was that like a particularly stressful, I imagine, experience finally going through those results?
Oh, yeah. I mean, it's very stressful. So like I said, I just knew that I couldn't keep beating my head against the wall, trying to identify soybean genes in the lipid pathway. And that, yeah, if this didn't work, I just had to move on to something else. And like I said, the scary part, I didn't even know what that something else was going to be. I just have to talk to other people in my department, other breeders or whoever, and just come up with something else. And that's a scary thought because I didn't even know what the alternative was going to be.
Absolutely. I think not having that backup plan sort of makes you work even harder.
Right.
I'm so glad to hear that that success worked out for you and your lab has really blossomed since then. You're doing amazing science, and I know that can motivate you to really invest even more time and energy into the science. But we love encouraging people to take a break, to give their minds a rest, and to think about something different. And you mentioned, of course, that you are working on a book on your own time in the beginning of our conversation, but I'd love to talk about books as well. Do you have a favorite book that you've read, Ralph, whether it's related to science or not, that you can recommend for me and our listeners to add to our own reading lists?
Well, if you haven't read it already, and a lot of people have because it's a very popular book, but I would recommend The Da Vinci Code by Dan Brown. I enjoyed the way it blended religious folklore with science and history, all within the context of a thrilling murder investigation. I mean, it's just a fun read. Also, in some sense, it provided an inspiration for what I'm trying to accomplish in the book I'm writing, where I'm merging prophecies in the book of Revelation with modern science to create an apocalyptic story that doesn't rely on supernatural events, all within the framework of what I hope will be perceived as a compelling storyline with interesting characters. And again, some of the whole thought or idea that you could blend religious concepts together with sort of science and history and all. A lot of that came from just reading the Da Vinci Code.
I like it. Well, excellent recommendation. We will add this to our list on our website. Listeners, if you haven't had a chance to read the Da Vinci Code yet, definitely put it on your lists. And we've talked about some of the variety of experiences that you've had as a scientist, the amazing work you've gotten to do. But I think another thing that people don't realize necessarily about careers in science are all of the places you get to go, whether it's traveling for research, for conferences, to work with collaborators. So, Ralph, do you have a favorite place that you've been to for science?
There's actually a couple of places that stand out. The first was a trip that I took to Guizhou province in China to visit a group of tobacco researchers who wanted to establish research collaborations with the tobacco genetics and breeding program at NC State. Now, Guizhou is in the interior southwest part of China and is a very mountainous region. Now, the trip was interesting from the beginning as we got there by taking a high-speed bullet train, which I'd never been on one of these before. But this train went through dozens of tunnels cut through the middle of mountains. I mean, it's quite the engineering feat, what it took to create this high-speed rail system through this mountainous region. And while we were there, we were able to go to the tallest waterfall in China, which is located there, and hike around that area. Now, the mountains in that region are very high in limestone, which makes them ideal for cave formation, and many of which these caves have their own underground river systems flowing throughout. And one day we were taken on a boat ride that snaked throughout this very long network of caves. That was really interesting. So a second trip that comes to mind was an international science conference that I attended in Berlin. And it occurred at a time that my son was in high school and he'd been studying German. So I decided to have him join me. It ended up being a great opportunity, not just to attend an excellent science conference, but also to check out the sights of Berlin with my son. One of the most memorable parts of that trip occurred when my son and I were just out at night exploring the city on our own, when we came upon this spectacular light show set to music that was being projected on the Brandenburg Gate. That was really impressive. It was a fun trip.
Oh, that sounds amazing. Now, do you speak German yourself?
No. And of course, just in high school, my son only knew a limited amount, but what amount he could do, say we're at a restaurant and all, or at a store that he could use. Hey, it's a lot of fun. It's a lot to be able to, in some way, apply the language that you've learned, even if it's to a small level.
Absolutely. And sometimes you just need a few key vocabulary words to make it through. So
Exactly.
Well, these sound like amazing travel experiences. And if our listeners are able to go to Berlin or to Guizhou, what do you recommend that they do or see while they're there?
Oh, definitely go to Guizhou to, again, visit the waterfall. It is the highest one in all of China. And I guess I'd also say check out some of the caves. I mean, some you can just hike through, where others, the only way you can approach them is get on a boat because they are full of water flowing through them. But again, those were the most unique things. And again, it's a mountainous region. There's a lot. I've been through all sorts of mountain ranges in the country, across the world, but these are rather unique. The rounded shape of them, it was just the type of mountain formations I really hadn't seen anywhere else. So even though it's not a typical tourist destination, Guizhou, most people haven't even heard of it. It's kind of off the beaten track of your normal tourist would go. I found this to be very, very interesting.
Oh, absolutely. I think some of those off the beaten path places are sometimes the most fun to go to because it's not overwhelmed with tourists. So
Right.
Well, it sounds like you've had some phenomenal opportunities to travel for science. And I think another thing about science that makes it a wonderful career are the people you get to work with. I think oftentimes there are still stereotypes out there about scientists being very serious and critical. And I know the research is important and often researchers are very passionate about the work that they do. But just some of the funniest and most creative and most amazing people I know are scientists. And I think you get to see this in sort of the lab traditions that develop just these quirky, funny memories that you share with colleagues. So Ralph, do you have an example of one of these human moments in science or something that goes against the stereotypes that people are used to seeing about scientists?
Oh, definitely. And for this question, I'm going to go back to my graduate student days, because during that time, a professor from nearby Duke University, he arranged to do a sabbatical in the lab of my PhD mentor for a year. He was one of the funniest people that I'd ever met, and he's a real prankster. He loved pulling practical jokes. And once he started them up, it kind of became contagious, and everyone in the lab began engaging in these pranks. One of these, for example, involved a jar of glass beads that was in the lab. And I can't even remember what the legitimate purpose of these beads were, but it became kind of a game of who could trick someone else into spilling them all over. So, for example, there'd be like a roll of paper towels at the end of the lab bench. And when no one is looking, someone would fill the tube up half full with these glass beads. So the next time someone would come grab the paper towels, the beads would go spilling all over the bench and the floor. And of course, the victim would be obligated to pick them up. Of course, whoever perpetrated it would never identify themselves. But we'd end up doing things like cutting out the bottom of a box of Kim wipes or a box of gloves, sort of booby trapping them with the beads and putting them up on a shelf. So when the person came to take the box down, again, the beads would go bouncing all over the place. Another thing that this visiting scientist did was he'd take some lab chemicals and he used them to make a small amount of a contact explosive. He would put these under the wheel of someone's lab stool, so when they got in their chair and moved forward, it made a really loud pop.
Oh, wow.
Now, there are other examples I could give, but most of them, sort of similar to the last one, involve things that would kind of give safety officials fits, like the inappropriate uses of liquid nitrogen. So it's best I just leave with the examples that I said. But needless to say, that particular year was one of the most memorable of my career.
Oh, absolutely. I love this idea of almost like the scientific confetti with the glass beads. Like instead of opening your birthday card and having all the confetti fall out, it's every time you reach for something in the lab, you've got a shower of glass beads.
Exactly. And it really became a game of who is going to fall for the bead trick next.
Well, I love seeing this sense of humor, and I think it brings people together, just having something that you can all talk about and laugh about that's not the science for a change. And I think this is important because I think taking a step back and having the spirit of community in a lab is important for addressing some of these big unanswered questions in science. And so, Ralph, if we took away the things that normally hold you back, I know it's usually funding, staff, technology, feasibility, time, et cetera. We could go on forever. But if we took those barriers away, what is the one research question that you would want to answer?
With the recent work that I've just talked about, we've shown that it's possible for one to take one of the mitochondrial genes from the plant, convert it to a nuclear gene, then delete it from the mitochondrial genome. So the bigger question I would like to know the answer to is, could we do this to all of them? And if so, what would be the consequences? So the rationale for wanting to know this is this. It seems awfully inefficient for the cell to be maintaining this completely independent genome inside the mitochondria for the sake of maintaining expressing a very small handful of genes. The human mitochondrial genome, for example, contains only 13 protein encoding genes. Now, there are approximately 250 genes in the nucleus that exist purely to enable the replication, transcription, and translation of this small mitochondrial genome to service those 13 genes. So the cell is arguably investing a lot of energy and resources for the sake of a tiny number of genes compared to the tens of thousands of genes found in the nucleus. Now, in plants, if we were able to completely do away with the mitochondrial genome in plants, transfer them all to the nucleus, could that improve the efficiency of growth and development because we no longer have to expend energy toward maintaining this whole separate machinery of DNA replication, transcription, and translation? So that's the big question that I'd like to know the answer to is eliminating the mitochondrial genome technically achievable? And if so, could there be a growth advantage to the plant by doing so?
This is a fascinating question. I'm glad you brought it up for me and our listeners to ponder today. And I'd love to talk about advice next, Ralph, at the end of our conversation. So when you think back along the course of your career in your life, is there one piece of advice that somebody gave you at some point that really helped you that you can share with everybody today?
So I actually can't think of an example where someone has given me a very specific piece of advice that by following it had a significant impact on my career. But indirectly, however, I could say that the best advice I was given was to believe in yourself. And by indirect, I mean that, say, both of my parents, my father in particular, conveyed to me the attitude that I could be successful in whatever I put my mind to. I remember, say, talking with my father when I was in high school about various career options, and some of them would be, say, careers that there weren't a lot of or very difficult to get. He would say things like, remember, there's always room at the top. But the way he would say it, what he was really conveying, doing it in a manner that was saying, don't worry, whatever you're going to go into, you're going to be one of the best. And there will always be jobs for those who are the best at what they do. That was sort of the attitude of his comments. So science careers aren't easy. So to me, going into it, having been instilled with a sense of confidence was important, especially during the times when things don't work out well, as we've already discussed.
Absolutely. I think having this confidence, this belief that you can do it is really important in science. And sometimes just having someone tell you that you can do it can help, because coming up with that confidence yourself can be difficult.
No doubt.
Well, wonderful message to share with our listeners today, Ralph. Is there any last piece of advice or maybe a last note of inspiration that you would like to end our conversation on today?
Yeah. So when it comes to doing research, my advice would be just to try as many different things as you can possibly accommodate. And one of my departmental colleagues liked to use the phrase, even a blind pig can find an acorn once in a while if it roots around long enough. Now, biological systems are complex. And so anytime you go into a new research area or start a new project, you're going into it with incomplete information. Otherwise, it wouldn't be science, right? So projects can often fail because you weren't aware of a hidden biological roadblock or information that would have invalidated your original hypothesis if you'd known about it, but it didn't exist at the time. So just by trying a lot of different things, being that pig that just roots around looking for that acorn, is trying a lot of things. Eventually, some of them are going to pan out. I mentioned before that if I were to look back at all the projects that we started, there's more examples of ones that failed than ones that succeeded. But if you do enough of them, you'll have enough successes that your career as a whole can be successful. That would be my advice, is as much as you can accommodate, just try doing as many things as possible, exploring as many avenues as possible.
Absolutely. I think that is excellent advice, because I think you're completely right here. If you put all your eggs in one basket and that basket fails, you are in trouble. But if you've tried a lot of different things, kind of put out a lot of different feelers, I think that gives you a lot more flexibility and a lot more opportunities to adapt and kind of pivot and go whichever way the data is showing you is perhaps the most interesting or most impactful.
Right. Yes.
Well, I think that is such an important message for our listeners today, Ralph. We appreciate you sharing that. And if our listeners are curious and they want to learn more about you and all of the amazing work that you're doing, what is the best way for them to do so?
The best way to get in contact with me would be through my email address, which is ralph_dewey@ncsu.edu. And again, I'd be happy to hear from anyone who would like more information about what we're doing.
Fantastic. Well, listeners, definitely reach out, get in touch with Ralph if you have specific questions, and take some time to learn more about his work. And Ralph, it's been such a pleasure to have you with us today. We really appreciate you joining us.
Well, thank you for the invitation. I enjoy talking to science and talking about what we're doing to anyone who's willing to listen. So thanks for the opportunity to do so.
Well, it's been great to learn more about you and your work, and listeners, always wonderful to have you here with us as well. We hope you join us on another episode of People Behind the Science.